Hi students, I hope you guys are doing well and welcome to another session of practice problem set for NPTEL course Introduction to Developmental Biology. It's already 3 p.m. and what we will do is we'll wait for 5 more minutes for other students to join. Meanwhile, I'll keep admitting those who request to join this call. So the instructions would be similar as the first practice problem session. You guys need to turn off your mic and video chat. Meanwhile, I'm presenting and talking to you guys. You can communicate anything with me in the chat box, and we'll be having a two-way session in which we'll solve some practice problem questions, exemplar similar to your quiz. Yeah, and this is a recorded session, so all the recordings will be on the YouTube, and the slides will be provided to you via Google Drive. So I have been uploading these slides in a Google Drive folder, and NPTEL will mail you the link. You can also find it in the course tab. And it won't be any further problem. Another TA class happens for this same course on Mondays from my colleague Partha from IIT Hyderabad. So Saturdays and Mondays you might be having class, and we'll solve practice problems related to lecture week 3, which consists of axis specification in Drosophila and differential gene expressions. I hope uh, you are going through your lessons. If not, you could catch up in the weekend, and the practice problem session is a perfect place to revise your concepts and also have a taste of how to solve your quizzes and assignments, which will help you in the main exam. So let's wait for 5 more minutes so that more students can join, and we'll start at 3536. Fine. And all the students are again requested to turn off their mic and video chat. You can communicate with me via the comment box. For the smooth conduction of this session, this is necessary. I'll share my screen once some more people have joined. Let's wait for 5 minutes. I can do one thing for all these students who are attending this session. At the end of this session, when we have some time left, what I can do is I can copy paste the link of the YouTube link for the videos. I've made a complete playlist in which the whole 12 weeks lecture course will have 12 different problem solving sessions. So all the 12 different problem solving sessions will be under that playlist. So whatever sessions that we are having right now, you can access the slides, and the recordings of these sessions will be on YouTube. I'll put that link in the chat box at the end of this session, so you have to stick with me. Also, you will get emails from NPTEL team uh, the regarding the slides and the recordings, right? You can also visit the course tab and find out. Rahul, if you have something you can type in the chat box, you can, or if it's something long, you can speak up. You were raising a hand, right? I think it was Rahul. Hello. It's okay, you can post your query in the chat box. I'll be constantly reading the messages, so don't you worry. It's already 6 minutes past 3. We should start our problem solving session. So what I'll do is quickly lay down the instructions again. So all the students, uh, please turn off your mic and video chat for smooth conduction of this session. And do not worry, it will be a two-way session where I'll be posting some questions corresponding to lecture week 3. We are solving week 3 today, and you guys have to answer. You guys can select the correct answer, whatever you think is the correct answer, and post it in the chat box. And we'll discuss further as to why the correct answer is the correct answer. We'll understand the concept behind that. So it will be a good way of revision for your concepts, and people who are still with pending lectures, they can go and watch and solve their own questions. It will also help you in the actual exam and the quizzes that you are going to be graded on, right? So let me just start sharing my screen. I hope you can see my slides and you can see the slides changing. You can just let me know in the chat box, yes or no. Is my audio clear and can you see my slides? The slides are changing because sometimes on these online platforms there might be some technical glitches and network issues where students have reported that they could not see the slides. Good, great. So once again, I welcome you all to the third session corresponding to the problem practice assignment for Introduction to Developmental Biology course. And the course instructor is, as you all know, Professor Subramaniam from IIT Madras. My name is Ashutosh and I'm a PhD scholar at IIT Kanpur. And I, along with Partha, will be the TA for this course. So in this practice problem assignment, I'll post 10 questions, multiple choice correct questions, similar to what you face in the assignment or in the quiz. And there will be 4 or 5 options and you have to guess or you have to tell me which is the correct option. And then I'll reveal the correct answer, proceeding which we will understand the concepts behind the correct answer, right? So I expect all of the participants to answer. Do not feel shy, be confident. It's okay if your answer is wrong. We'll understand the concept and you'll get it right the next time, right? So I expect all the people to participate. I encourage everybody. So the first question is here. The first question is a true false based question. Which of the following is correct? Statement 1, a large fraction of complexity between different organisms can be attributed to differential splicing. It's a statement about differential splicing. And statement 2, differential splicing can result in a large number of mRNA transcripts and hence a large number of proteins being produced from a single gene. Right? These are the two statements and you have to find out which is true and which is false. Option A, statement 1 and 2 are true. Option B, statement 1 is true but statement 2 is false. Option C says statement 1 is false but statement 2 is true. And option D, both are false. I hope you are able to see the question clearly and I have read it out for you. You can take some time, let's take some time and ponder about it. Which of these statements are true or false? And you can let me know what do you think is the correct answer in the chat box. You can just type the options or A, B, C, D, whatever you think. Let's collect some answers from the students. Pushpanjay is answering option B. Okay, 1 is true and 2 is false. And what about others? See, people, no need to shy out. You take your time. You can just guess. If you are not clear with the concepts, we'll get our concepts clear in the following slides, right? We'll see what is differential splicing and what these two statements mean. You can quickly type what you think is the correct answer to this question. Which of the following is the correct statement? Okay, okay. Seems like nobody else is able to figure out. Now let's see what is the correct answer to this question. So option A is the correct answer to this question. Both these statements 1 and 2 are true. So large fraction of complexity between different organisms can be attributed to differential splicing. Differential splicing, in the other words, alternative splicing. And differential splicing can result in large number of mRNA and hence large number of proteins from a single gene, right? So let's learn a little bit about alternative splicing and how it induces complexity between organisms. So this here you see is an example where an ancestral gene A has three exons. You can see here the gray regions, two gray regions and a black red region are exons joined by introns. So exons are the coding sequences and introns are non-coding sequences. Now this is the ancestral gene A. Now alternative splicing can happen two ways, in which all these three exons are joined together to form ancestral isoform 2, or the first exon and third exon can be joined together to give an ancestral isoform 1. So now you see from a single gene, two different types of exon joining can happen due to alternative splicing, which can result in different mRNA and protein structure. So now, if this ancestral gene A is present in a common ancestor, two other organisms derived from the ancestral A will have two different tissues specific proteins. So let's say an ancestor gave rise to a worm and a human. The same gene, the same protein could give rise to tissues, let's say nervous tissues, in worm as well as in human. But those proteins will be derived from the same ancestral gene via alternative splicing. So by enabling the creation of novel protein functions, alternative splicing contributes to the diversity and adaptability of a species. So a species increases its adaptability via alternative splicing. From the ancestral form, we can do alternative splicing to render a protein different protein function than from the other, because the organism might be in a different environment offering different advantages to adaptability. And splicing contributes significantly to the complexity of different organisms by generating diverse proteins isoform from a single gene. So that is what we studied here. From a single gene we can have diverse protein isoforms, and these proteins will be from the similar family because they will have certain sequences which are conserved in both the proteins, and these diverse protein allows for a tissue specific expression, facilitating evolutionary adaptation, enabling fine tuned regulatory mechanisms, and even influencing disease outcomes. So tissue specific expression of different isoforms, different genes, a same gene giving rise to different proteins gives rise to complexity in different organisms. So if you see, as Professor Subramaniam has mentioned in his lecture videos, a C. elegans worm and a human have almost similar kind of genes, but the protein array of humans is quite diverse from that of a C. elegans due to alternative splicing. Right? So this explains our statement 1. Now for the statement 2, Differential splicing can result in large number of mRNA transcripts and hence a large number of protein being produced from a single gene. So for this statement, here is a representative diagram which will ease up your explanation. In this DNA segment, we have our transcription start site here, and there are few exons. There are 5 exons here. Exon 1, 2, 3, 4 and 5 color coded. Blue is exon 1, green, red, cyan and orange. Now when it transcribes into the RNA, so the exons will be transcribed as well as the introns before the RNA is processed. Now when the RNA will undergo post-transcriptional processing into mRNA, so there will be alternative splicing in which different permutation combinations can happen where exon 1, so this is one type of mRNA in which all the exons are joined together, exon 1 with 2, 3, 4, 5 in an order. This translates to protein A, while in alternative splicing of another isoform, we see the red exon 3 is now omitted, exon 1, 2, then 2 is joined to exon 4 with 5, and in this isoform, exon 4 is skipped, exon 1, 2, 3 and 5 are joined. So this translates to different proteins. You see protein A, protein B and protein C are different. And you can figure out that this is the whole protein and these are isoforms of the protein. So here we see that a single gene can give rise to 3 different mRNAs, which can lead to production of 3 different proteins from a single gene. So an example of 3 different proteins produced from a single gene due to 3 different mRNAs produced by alternative splicing. So that proves statement 2 is also correct and it's true. That's why the option 1 is the correct answer to this question. Both the statements are true. Statement 1 and 2 are true. So organism complexity is brought about by differential splicing, and one gene can produce many mRNA and many proteins via alternative splicing or differential splicing. I hope the explanation is clear to the students. You can quickly let me know in the chat box with a yes or no. So you guys need to understand the basic concept behind alternative splicing and how it influences the organism. I hope you are a bit clearer about the concepts. Is it clear to the students? You can just let me know with a yes or no. Yes, great, great. Now let's move on to the next question. So the next question we have is a match the following question. On the left hand column and the right hand column we have one exact match. So on the left hand column we have maskin, casein mRNA, misspliced RNA and dscan. And on the right hand side we have dendrite branching, muscle hypertrophy, prolactin, circularized oocyte mRNA. And your options are option A 1A 2D 3C 4B, option B 1D 2A 3B 4C, option C 1D 2C 3B 4A and option D 1A 2C 3D 4B. So the question is before you. You have to match the left column correctly with the right column. Now students, what do you think is the correct answer? Let me know in the chat box with a yes with your options. Which of the options do you think is the correct one? Okay, Shubhangi has answered option D 1A 2C 3D 4B. Okay, let's see. Let's collect some more answers. A couple more answers and we'll look at the solution. Come on students, I'll encourage everyone to answer. These are very simple multi-choice questions, the way you are going to face in your final exams as well as your quiz and assignments. And if you have been through the lectures, you will be able to answer it. It's no difficulty. Even if you do not know the answer, you can just guess according to your logic or whatever knowledge you can recall. we will anyway move on to see what is the correct answer and understand the concept behind it. So I'll encourage all the students to at least try answering in the comment box. You just have to write your options. Let's collect a couple more answers and then I'll reveal the correct answer. So Shubhangi has answered option D. Let's see what other students think is the correct answer. Okay, Pushpanjay has answered option B. Yeah, let's not delay it any further and reveal what is the correct answer to this question. So the correct answer to this question is option C. 1D, 2C, 3B and 4A. This is the correct answer to this question. So maskin is circularized oocyte mRNA. Casein mRNA will match with prolactin, misspliced RNA with muscle hypertrophy, and DSCAM with dendrite branching. Okay, so this is the correct answer. Now let's move on with one by one what are the left and right match columns. So this is about maskin. So in the oocyte, there are maternal RNAs which are already present in the cytoplasm of the oocyte, which are translationally dormant because they do not want to get translated at the wrong time. Only when there is maturation signal, these mRNAs will be translated. So the mRNAs are made and are present in the oocyte already, but the mRNA that are present are in a circularized form because the 5' cap of mRNA is bound by a protein called 4E, and the poly A tail is bound by another protein called CPEB, and maskin is a protein which binds the 5' cap and CPEB protein and prevents the recruitment of initiation factors, making the mRNA translationally dormant. Such mRNAs attain the circular structure. So maskin will bind these proteins which are holding together the 5' cap as well as the 3' poly A tail, and binding of maskin and these proteins will inhibit the initiation of translation because in the presence of maskin, the initiation factors cannot be recruited to the mRNA. Now when there is a maturation signal such as prolactin hormone, so when the female receives prolactin hormone, now the oocyte is ready for maturation. What happens is a series of events follow. This CPEB protein which is binding to the poly A tail is now phosphorylated, and poly A tail is extended via other different proteins such as poly A polymerase. There is addition of poly A tail to the existing poly A tail, and maskin therefore cannot bind to phosphorylated CPEB and the cap protein and will stop the inhibition, while 5' cap protein 4E can recruit 4G and other proteins which can recruit the ribosomal subunit and poly A binding protein, and thus translation initiation can begin. So translation will be active only when maskin is not binding the cap and CPB protein when CPB is phosphorylated. So maskin binding to the cap and CPB will create a circularized mRNA structure which is found in the oocyte. So maskin on 1 will match D, circularized oocyte mRNA. Now let us see some other options. So this is an example that Professor Subramaniam has clearly explained in his lecture videos. Misspliced mRNA of myostatin gene leads to inclusion of intronic sequences harboring stop codons. So missplicing of a myostatin gene causes muscle hypertrophy or mighty mouse. So this is an example of mighty mouse in which the muscle cells are bigger, muscle fibers are bigger, and what happens is that The normal myostatin protein has a termination codon or stop codon in an intronic sequences. So generally intronic sequences do not are not present on the mRNA because they'll be spliced out. They are not coding for any protein. So any presence of stop codon or absence of stop codon doesn't matter because the intronic sequences will not be coded. But what happens in if a mutation occurs in the intronic sequence, the GTAAGT that we are seeing here, one of the residues of G is mutated to A adenine, and this A acts now as a splice donor site. So splice donor and splice acceptor of exon 2, which now due to alternative splicing, the part of intron is now encoded as an exon into the mRNA. So now the mRNA has a premature stop codon which was earlier present in the intron that was getting excluded. Now because of this mutation, the splice donor and acceptor sites have changed, which leads to incorporation of this mutation in the coding sequence. Now because of this stop codon, there is a truncation in the protein myostatin. So the protein will not proceed further this stop codon, and there is a non-functional protein which leads to muscle hypertrophy. So this is how mutant mice looks like with a mutation with a missplice of myostatin gene. This leads to truncated myostatin protein production which results in muscle hypertrophy. So misspliced RNA will match myostatin gene. Misspliced RNA, muscle hypertrophy. This will be the perfect match. Then let us study about DSCAM and what protein it is. So there is a phenomenon called neuronal self-avoidance, which is an important property of neurons, which consists in the tendency of branches, which are dendrites and axons, arising from a single cell body to turn away from one another. So it says that the branches from a same cell body, from a same neuron, will not form connections with each other. Because we do not want the branches of the same neuron forming connections. The neuron should connect to a new neuron so as to relay the information. Otherwise, the electrical signal will get jumbled up and the neurons will all connect to each other. So how does the neuron know that this is my own dendrite and I do not have to connect to them? This is due to the presence of the protein DSCAM. So in Drosophila, the protein DSCAM present plays an important role in controlling cell intrinsic aspects of dendritic branching and guidance. So similar type of DSCAM molecules, if present, so similar kind of DSCAM pattern will be present in a dendrite arising from same neuron. So those dendrite will repulse each other and thereby perform self-avoidance. So they will not form connections with self, they will form connections with other new neurons. But it has been seen that when there is a DSCAM mutant Drosophila fly, so there are no proteins here now, and due to no proteins here, there is no repulsion, due to which self crossing will happen and there will be overlap of self junction neurons. So this is the disease phenotype due to absence of DSCAM protein. So here DSCAM will be matched correctly to dendritic branching. So then our correct answer will be maskin to circularized oocyte mRNA 1 to D, DSCAM to dendritic branching 4 to A, misspliced RNA to muscle hypertrophy 3 to B, and casein mRNA to prolactin 2 to C. So this will be the correct answer. So I hope the explanation is clear to the students, and I have tried to keep the slides really easy so that anybody will be able to understand it. Is it clear to the students? You can just let me know with a yes or no. You can just let me know in the chat box. Is it clear to the students? Good, good, great. Now let's move on to the next question. Question number 3. It's a fill in the blanks type question. In the dash precursor cell dash levels of MIR 181 gives rise to both B cells and T cells, while artificial introduction of dash levels of MIR 181 gives rise to larger number of B cells. Okay, so this question is related to microRNA. I'll give you a short hint. MIR 181 is a microRNA. MIR stands for microRNA. Now you have to answer which options fits the fill in the blanks. Option A, lymphoid high and low. Option B, lymphoid low and high. Option C, myeloid high and low. Option D, myeloid low and high. Now which of these is a precursor cell, and what levels of microRNA 181 gives rise to both B cells and T cells, while artificial introduction of dash levels will give rise to large number of B cells. Come on students, let us collect some answers. You can take a minute to read the question carefully and look at the options and mention your answers in the comment box, whatever you think is the correct option. And after a minute, I'll reveal the correct answer and we'll move on to the explanation. I'll encourage all of the students to answer. This will easily revise your lessons and your concepts. So I would expect all the students to answer. Rahul has answered option A. Okay, good Rahul. And can we have some other answers from the students? Ashwin, Simontini, and Shubhangi, Pushpanjay. Just try to answer. It's okay if you are not able to give a correct answer. Okay, Pushpanjay has answered option C. Okay, let's not delay it anymore, and I'll reveal the correct answer now. So the correct answer is option B. So option B is the correct answer. In the lymphoid precursor cell, low levels of microRNA 181 gives rise to both B cells and T cells, while the artificial introduction of high levels of microRNA 181 gives rise to large number of B cells. So this is the correct answer. So always a trick to remember is B cells and T cells are lymphocytes. We call them B lymphocytes and T lymphocytes. So if you now eliminate some options, lymphocytes always come from lymphoid precursor cell or lymphoid progenitors. So it can never be myeloid progenitors because B and T cells are lymphocytes. So you can eliminate option C and D right away, and then you have to understand, you have to recall what are the levels of and what is the role of MIR 181 in production of B and T cells, right? So in summarizing, the lymphoid precursor can generate B or T cells. So lymphoid precursor cell, low MIR 181. When the level of microRNA 181 is low, it can generate B cell MIR 181 and T cell with MIR 223, no MIR 181. So in absence of MIR 181, MIR 223 will produce T cell, and no MIR 2 to 3 will produce B cell. So when low MIR 181, equal amount of B and T cell can be produced from the lymphoid precursor. But if we virally activate MIR 181, means we use a retrovirus to introduce MIR 181 into the lymphoid precursor cell, we have a larger number of B cells and number of T cells reduced, which can be very obvious because MIR 181 promotes the proliferation of B cells or the production of B cells from the lymphoid precursor cells, while presence of MIR 181 will decrease the number of T cells. So if we externally or we experimentally increase the quantity of MIR 181 via viral infection, retrovirus carrying the gene for MIR 181, then we can see there is an increase of B cells number. So the correct answer would be lymphoid precursor cell, low levels of MIR 181 giving rise both B and T cells, and high level will give rise to larger number of B cells. I hope this is clear. Now let us quickly move on to look at how does these MIRs or microRNAs perform their function and what function do they perform. So in a normal MIR gene, so there are genes which codes for microRNAs. These microRNAs do not give rise to proteins. So not all RNAs in the cell are translated into proteins. Many RNAs have regulatory roles, and there are many species of other RNAs like ribosomal RNA, which constitutes the ribosomal subunit, tRNA or transfer RNA, which is involved in translation. So these are all RNAs, but only the mRNA or messenger RNA is what is getting translated into proteins. So messenger RNA is the one which is transcribed, which is spliced and then translated into protein. So miRNA is another form of RNA which is not translated into the protein, and miRNA, microRNA, controls gene expression mainly by binding with messenger RNA. So it is a regulator of gene expression in the cell cytoplasm. So what does it do? It binds with the messenger RNA, and instead of being translated quickly into a protein, now the mRNA, which is bound by the microRNA, instead of that being translated quickly into a protein, the marked mRNA will either be destroyed and its component recycled or will be preserved and translated later. So there can be different fates of the mRNA when it is bound by the microRNA. It can be destroyed, the components of the mRNA can be recycled, or it will be preserved and translated later whenever the time comes or whenever the signal arises, need arises. So how are these miRNA produced microRNA? So there are certain genes which can produce microRNA. Transcription factors will transcribe via RNA pol 2 polymerase 2 and generating the 7-methyl guanosine capped RNA with poly A tail. So this is a microRNA, looks exactly like the mRNA, but it does not translate. But what does it function in? So there, the in the microRNA, once it is generated, certain endonucleases cut down the RNA into hairpin structures called pri-miRNA, which is now processed by an enzyme complex called DGCR8. So DROSA DGCR8 is an endonuclease or a DNA cutting enzyme, sorry, nucleic acid cutting enzyme, which will now cut these into the hairpin structures, which is called pre-miRNA. Now this pre-miRNA will exit the nucleus. Pre-miRNA can then be acted upon by dicer protein. Now dicer protein will remove the loop region and the hairpin region and load the pre-miRNA into RISC complex. So RISC is a complex called RNA induced silencing complex. Now the RISC complex can scan the genome for the complementarity of this microRNA with the genome. Wherever the mRNA it finds its complementarity, so RISC will go and can perform many different functions such as inhibition of translation, degradation of the mRNA, or de-adenylation of the mRNA. So these are one of the many functions that microRNA can perform. So microRNA can regulate the gene expression via binding with messenger RNA. So first they will be processed by DROSA complex, then exit the nucleus processed by dicer complex, enter the risk complex, RNA induced silencing complex, and then can scan the cytoplasm for complementarity and induce degradation, deadenylation, or destabilization of the messenger RNA. So this is how is a general principle microRNAs work, and here we saw the influence of microRNA 181, which promotes B cell proliferation from lymphoid precursor cell, and in the absence of microRNA 181, T cell production can increase. I hope this is clear to the students. We learned a bit about microRNA and the lymphoid precursor cell. Is it clear to the students? You can quickly let me know. A yes or no in the comment section would help. Is it clear to the students and did you understand it? Great. Good. Okay, so far so good. Let's move on to the next question. We'll keep moving. So answers question 4 and 5 based on the following. Questions 4 and 5 will be linked, and based on the following you have to answer both the questions. Oscar mRNA is localized to the dashed end of the Drosophila oocyte. Oscar mRNA localization in the Drosophila oocyte. Which end? Is it the anterior end or the posterior end? Students, please answer. What do you think? Which side of the Drosophila oocyte is Oscar mRNA localized? I would expect some answers at least. It's a two option question. It can either be anterior or posterior. You have to recall the anterior posterior polarity and axis patterning in Drosophila. Where do you think is Oscar mRNA localization? Okay, Pushpanjay is answering anterior. Simontini is saying posterior. Rahul is also saying posterior. And let me reveal the answer. So option B, posterior, is the correct answer. Simontini and Rahul have answered correctly. And thanks for the efforts, Pushpanjay. But Oscar mRNA localization is towards the posterior end of the Drosophila oocyte. So here if we see in this schematic, this is a time-dependent developmental stage from formation of a zygote, fertilization, and the development of the egg of the Drosophila. So anterior to posterior axis. So after fertilization, we have a nerve cell and a oocyte. So before fertilization, so this is the maternal, not fertilization. I'm sorry. This is the maternal oocyte developing. So there are certain nerve cells which provide nourishment to the oocyte, and this entire nerve cell oocyte combination is enclosed in what is known as follicle cells. Now what happens? Oscar mRNA is localized to the posterior end. So this will become the future posterior end, and here you can see mRNA in situ hybridization data. So mRNA in situ hybridization data for says that wherever the RNA is produced, we have a labeled antisense RNA which will recognize the Oscar RNA, and we can detect the antisense RNA with antibodies. So in this way we can determine what is the spatial context of RNA localization. So here we see that Oscar mRNA, when an antisense probe used against Oscar mRNA, it shows future posterior size localization. Oscar localizes to the posterior pole via bicoid localizes to the anterior pole, future anterior pole. And question 5, which is a follow-up of that question, how is this localization brought about? This localization in sense, how is the localization of Oscar to the posterior pole brought about? And there are five options this time. They are trapped by nanos mRNA present at the end, option A. Option B, they are trapped by nanos mRNA present at the opposite end. Option C, they are transported by kinesin in the minus to plus direction. Option D, they are transported by dynein to the plus to minus direction. And option E, they are degraded everywhere except at the end. Now, which of these mechanisms do you think will localize the Oscar mRNA at the future posterior part of the Drosophila embryo? Take time to read through all the options and then it will be easier for you guys to find out which is the correct answer. I'll be waiting in the comment box for some answers. Whichever you think is the correct answer, you can just let me know, A, B, C or D or E. What is the mechanism in which Oscar mRNA is localized to the posterior pole? Guys, is the question difficult for you guys? You can just guess any answer if you think uh, whichever is the correct option. What is the process of localization of this Oscar mRNA? Okay, Pushpanjay has answered option B. And others, do you think you can answer this? I'll encourage everyone to answer. See, you are not going to be marked or judged or punished for all this. You should participate more in these problem sessions so that you can take back the maximum benefit, right? Because I'm here to help you with your assignments and your quizzes and also with revising and learning your concepts. So you should fully utilize this time. Rahul has also answered option B. Okay, let me reveal the correct answer to this question. The correct answer to this question is option C. Oscar mRNA is transported by kinesin in the minus to plus direction. Guys, this is the correct answer, option C. You guys were confused about the nanos mRNA, but we'll see if in the upcoming slides what is the relationship between nanos and Oscar mRNA. Okay, so that it will clear your confusion for once and for all. So the mechanism in which Oscar mRNA is localized is via transportation by kinesin in the minus to plus direction. Now let us learn a little about the anterior posterior polarity in Drosophila embryo. So here in a mature egg cell, you see there are nerve cells which nourish a single oocyte, and there are uncommitted polar follicular cells. Now certain cells in the pole which are neighboring the oocyte will become the posterior pole and the other cells will become the anterior end at here. So what happens in the early stages? The oocyte moves into the posterior region of the egg chamber while nerve cells fill the anterior portion. So this, guys, you could know that the side to which oocyte is closer will form the posterior end and the side to which nerve cells are closer will form the anterior end. The oocyte nucleus moves towards the terminal follicle cells and synthesizes Gherkin protein. So nucleus of the oocyte synthesizes Gherkin protein. So the green protein that you see here, green colored dots, are the Gherkin protein. And what the terminal follicle cells do, the follicle cells which are surrounding the oocyte will express the receptor for the Gherkin protein, which is called torpedo, the receptor for Gherkin. So you can see how the oocyte and terminal follicular cells harmonize. The oocyte nucleus produces the ligand, which is called the Gherkin protein, and the terminal follicle cells produce the receptor. So they express the receptor on the surface as if to receive the signal which is coming from the oocyte nucleus. So what happens when Gherkin binds the torpedo, or which is the receptor for Gherkin? When Gherkin binds to torpedo, terminal follicle cells differentiate into posterior follicle cells and synthesize a molecule that activates protein kinase A in the egg. So what will happen when the Gherkin binds to the torpedo? These terminal follicular cells will now take the identity of posterior follicle cells. And what they will do, they will synthesize what is called a protein kinase A. So you remember this part? Oocyte will make Gherkin protein. The terminal follicular cells which are near to the oocyte will express the receptor which is torpedo. Now Gherkin binds to torpedo. The identity of terminal follicular cells change to posterior follicular cells, and these now express and produce a protein kinase A. Now when protein kinase A is produced, you can follow the diagram here. Now these cells are posterior cells. Protein kinase A orients the microtubules such that the growing ends are at the posterior, depicted in panel D. So what protein kinase A will do, it will rearrange the microtubules in such a way that the plus ends of microtubule will be towards the posterior end, and the minus end of microtubule will be toward the anterior end. Right? Now there are two RNAs which come into play. Bicoid mRNA binds to dynein, a minus directed motoprotein associated with non-growing end of microtubules. Dynein moves the bicoid mRNA to the anterior end of the egg. So here you can see the bicoid mRNA is taken by the dynein protein. So dynein is a motoprotein which moves towards the minus end of the microtubule. Whereas on the other hand, Oscar mRNA becomes complexed to kinesin, which is the contrary protein which can travel cargoes to the plus end of the microtubules, a plus directed motoprotein that moves it towards the growing end of microtubule at the posterior region where Oscar protein can bind nanos mRNA. So what happens? Oscar sits on kinesin and moves to the plus end of the microtubules, whereas bicoid sits on dynein and moves to the minus end. Now the Oscar RNA, when reaches the posterior follicular cell side, it translates. So the RNA is translationally dormant while moving on the kinesin, while when it reaches the oocyte, posterior side of the oocyte, it translates into Oscar protein. Now Oscar protein acts as a trap to bind nanos mRNA. So the option B was wrong because option B said the Oscar mRNA is trapped by the nanos mRNA present at the opposite end. Whereas what will be the correct statement is Oscar protein traps nanos mRNA at the posterior end. That is what is actually happening. But the correct answer to this question will be nanos mRNA sits on kinesin motor and moves from minus to plus end directed. So you can see here they are plus end directed motors, and Oscar will sit on kinesin and with the help of that will move to the posterior side. This is a brief, very brief, short overview of localization or generation of polarity in the Drosophila egg. I hope it's clear to the students how what is Oscar protein, Oscar mRNA, and how does it localize to the posterior end. Is it clear to the students? Just let me know with a yes or no. So we just solved two questions based on Oscar mRNA and Drosophila axis patterning. anterior to posterior polarity. Yes, yes, great, cool. Now let's move to the next question. Question number 6. Determine whether the statement holds true always. Lower the recombination frequency between two loci, lesser the physical distance between them. Now this is a question relating to physical distance between two loci and the recombination frequency. I'll give you a subtle hint. This is based on the chromosomal theory of linkage, which was proposed by Thomas Hunt Morgan. T.H. Morgan was a great scientist known especially for his chromosomal theory of linkage. So if you are aware of that theory, you will be able to answer this. Again, students, you have to read the question very carefully. Lower the recombination frequency between two loci, lesser the physical distance between them. Is it true or is it false? Very simple question. Two options, true or false. I am expecting some people to answer. Read the statement carefully and just let me know what you think about the answer in the comment box. Lower the recombination frequency between two loci, lesser the physical distance between them. These terms could be quite confusing, but these are very basic and fundamental, and you have to have your concepts right uh, if you want to do good in developmental biology. Okay, Rahul has answered B, false, and can we have couple more answers? Pushpanja is also saying B. Good. Ashwin and Simontini, are you able to figure out the answer? You can just let me know what you think is the correct answer. It's okay, you do not need to shy out. Anyways, you just have to type in the comment section, right? Okay, I think we should move forward. Now let us see, let me reveal what is the correct answer to this question. Correct answer is option B, false, and both Pushpanja and Rahul as well as Simontini has answered now correctly. Very good. So lower the recombination frequency between two loci. So between two loci in a chromosome, if the recombination frequency is low, lesser the physical distance between them. It's always not true, it's false. So lesser the physical distance, recombination frequency is not always low. Now let us understand the basis of recombination, DNA recombination. DNA recombination, also known as genetic recombination, refers to the process by which DNA molecules exchange or combine genetic information, leading to creation of new combination of genes or alleles. So we see that DNA molecules, when two DNA molecules come close together with homologous sequences, they can exchange and combine genetic information amongst themselves, which can lead to various combinations, mutations, and divergence of genes and formation of new alleles. So this process of DNA recombination occurs naturally during various biological events such as meiosis. Meiosis is the reduction division which happens in the germ line to produce haploid gametes. So diploid organisms like humans produce haploid gametes via reduction divisions. So humans have 23 pairs of chromosome, 46 chromosomes, but in meiosis, which produces a single haploid sperm, will have 23 chromosomes, just 23. And during that meiosis, there is an event called crossing over, which is mentioned here very beautifully, in which two homologous chromosomes align together and strands of homologous chromosome can undergo genetic recombination, and exchange of genetic material happens between them to give a recombined chromosomes. Now these can form the basis of gametes. That's how variation, the DNA recombination is a major source of variation and a driver of evolution in animals. DNA recombination is crucial for increasing genetic diversity within populations and is a driving force behind evolution. So this is just what we discussed as of now. DNA recombination is crucial because it increases the genetic diversity via genetic recombination, exchange of DNA material between neighboring chromosomes and similar sequences. It increases the genetic diversity, and within a population of animals, genetic recombination brings out the variation and is also a driving force behind evolution of species. So meiosis is a natural biological phenomena in which DNA recombination occurs very frequently in the prophase 1 of meiosis 1. Right? And now let us know a little about linked and unlinked genes. So as I was mentioning, the chromosomal theory of linkage proposed by Thomas Hunt Morgan says that tightly linked genes occur close together on a chromosome and show low recombination frequency, and they are likely to be inherited together. So two genes or a group of genes, here gene 2, 3, 4, are known as a linkage group because they are present very close nearby in a chromosome and therefore have a high chance of getting propagated together in the gamete. So if there is a crossing over event, the crossing over will not be, the crossing over frequency within gene 3 and 4 and 2 and 3 will be very less, and it is more likely that 2, 3, 4 are inherited as a group when the gamete formation happens, right? So lower recombination frequency implies greater physical distance between them. Recombination frequency is the frequency with which a single chromosomal crossover will take place between two genes during meiosis. And a centimorgan is a unit that describes a recombination frequency of 1%. So a centimorgan is a unit for depicting 1% recombination frequency. I hope it's clear to the students. I think I'll have to modify a little bit on this statement. Actually, it's the reverse. So lower recombination frequency. So the less likely two genes will recombine means lesser physical distance. So it's generally the case, but the answer to this question will be same because there are certain cases in which genes which are located farther can have lower recombination frequency due to many other reasons. Lower the recombination frequency between two loci, lesser the physical distance between them does not always holds true, but generally the case is if two genes are tightly linked, they are closely associated with each other and the probability of recombination between them is lower. So lesser the physical distance, lower the recombination frequency. This is correct, but it is not always true, does not hold true always. That's why the false answer is correct. So the explanation to this, I would I would correct myself. Lower the recombination frequency implies lesser physical distance. This is correct. Now let's move on to the next question, question 7. So this is another question depending on recombination frequencies. Consider four genes A, B, C, and D. Using genetic crosses, the recombination frequency for different pairs were found to be as follows: C and A 17%, D and B 24%, B and A 3%, and C and D 2%. Now, what is the most likely the positions of these four genes on chromosome? You guys might need a pen paper for this. You might have to visualize and draw a diagram and show the position of these genes depending on the recombination frequency. Just previously we learned the rule that closer the genes are, lesser is the recombination frequency. Now I have given you the biggest hint. So now let us wait for some answers. What do you think is the likelihood of most feasible arrangement of these four genes A, B, C, D? C and A has 17% recombination frequency. D and B have 24% recombination frequency, while A and B have 3% and C and D have 2%. The options are B, C, A, D, option B, D, C, A, B, option C, B, D, A, C, and option D, C, D, B, A. And option 5 or E, they are not all on the same chromosome. Now guys, just let me know what is the correct answer. It will be a fun exercise if you try to solve it yourself. Take a pen and paper and just sketch out what will be the arrangement of these four genes A, B, C, D based on their recombinant frequencies. Pushpanjay has a problem, I think. Sir, I can't fix this. Pushpanjay, can you, what problem are you facing? Is it the internet or are you talking about the question? If you're not able to solve the question, it's fine. Just take a guess or just take a hunch. What do you think is most appropriate out of these options? If there were a question before you like this in an exam, you could just take a hunch. Based on the recombination frequency, I gave you the biggest hint, right, guys? The higher recombination frequency will occur when genes are farther apart, and low recombination frequency will occur when genes are close apart. So now what do you think will be the arrangement of ABCD of these four genes? Just try out. Let us wait for a minute and collect some answers. Okay, Pushpanjay has answered option D, C, D, B, A. Okay, let's see whether that's correct or not. And others, are you able to follow the question? I hope you will be able to answer it. I think other students are not able to solve this question. Any guesses you any guesses you want to make? on these options? Rahul, Simontini, and Ashwin. Oh, Rahul has left for some reason. Okay, let's not delay it any further, and let me reveal the correct answer. The correct answer to this question is option B, D, C, and A and B. So this is the arrangement of four genes on the chromosome or on any gene segment based on their recombination frequencies. What is the most likely positions of these four genes on a chromosome? C and A 17%, D and B 24%, B and A 3%, C and D 2%. Now if you look at it, the diagram will look somewhat like this. So this is the important uh, theory that we learned. Recombination frequency is inversely proportional to the distance. So higher the distance between two genes, so lesser the distance, lesser the recombination. I'm sorry. Just let me rectify this quickly. Yeah. So if there is a high recombination frequency, that means two genes are very distant. That's how they are able to recombine so efficiently. So we see that A, B have recombination frequency of 3%. That says that A and B must be close by. C and D have a recombination frequency of 2%. That means C and D are also close by. Now the arrangement of A, B, C, D could be either one of these. But this is the final arrangement because C and A have a recombination frequency of 17%. So they might be closer than D and B, which is recombination frequency of 24%. So this is how we came to the answer of this arrangement DCAB. Lesser the distance between them. So you see, if D and C, if you imagine this as a gene arrangement on a chromosome, D and C are tightly linked. They are closely associated. The distance between them is very less. That's why recombination frequency is so low that there cannot be a crossover between D and C exchanging the C to another chromosome. So D and C are likely to inherit together into next generation. Similarly as A and B, because A and B have only 3% recombination frequency, they must be close together to have this less of recombinant frequency, and they are more likely to be inherited together in the offspring. Whereas recombination frequency of 24% and 17%, this shows that crossover can happen in between C and A and D and B with higher frequency. So there are less likely chance that D and B will be inherited together. There might be crossover events which will interchange genetic information between the D gene and the B gene. So this is the final arrangement and answer to question number 7. DC recombination frequency 2%, AB is 3%, AC is 17%, and BD is 24%. According to this is the correct answer. I hope students are able to understand. Is it clear to the students? Is it clear to the students this arrangement based on distance and recombination frequency? It's very simple. If you imagine the crossing over event in meiosis, two loci or two genes associated very closely will inherit together and there will be less chance of recombination between them. and genes which are farther apart will have higher recombination frequency. Okay, yes, good. Let's move on to the next question. Question number 8. Data on a particular trait was collected from a population. The variation in this data was found to form a bell curve. This particular trait then is most likely to follow which of the following properties? Option A, incomplete dominance. Option B, pleiotropy. Option C, polygenic inheritance. Option D, complete dominance. Option E, environmental determination. Now you have to recall your concepts about incomplete dominance, pleiotropy, polygenic inheritance, dominance and recessivity, and then it will be easy to answer this question. Let me know in the chat box which you think is the correct answer. Simontini answers C, polygenic inheritance. Okay, good. And let us gather some couple more answers. Then we study about the concepts. Rahul has answered option D, complete dominance. So we have to remember the characteristic of this question is the variation in this data was found to form a bell curve. And then this particular trait, what could be this trait which follows a bell curve distribution? Simontini answered C and Rahul has answered D. Pushpanjay is also saying option D. Okay, okay. Now let us look at what is the correct answer. So the correct answer is C, polygenic inheritance. Simontini is correct. She has answered correctly. Polygenic inheritance is the property which shows a bell curve data. So complete dominance is not the answer, unfortunately. Now let us learn a little bit about what is polygenic inheritance and how does it show a bell curve data. So if you see here, polygenic, the term itself means poly and genes, many genes. So polygenic inheritance or traits are traits which are controlled by many genes. Polygenic traits are controlled by more than one gene. So till now we have learned one gene, one trait hypothesis. Like uh, if there is a gene for white eye, then the Drosophila will have white eye. If there is a gene for red eye, it will be red eye. But some genes are not so simple to study. There can be polygenic inheritance in which many genes control many, sorry, polygenic traits. Many genes control a single trait, and pleiotropism in which single gene controls many traits. So these are two polar opposites. Many genes, one trait, polygenic. One gene, many traits, pleiotropic. Right? So instead, several genes contribute to the final phenotype of a given trait. Polygenic traits are controlled by more than one gene. Instead, several genes contribute to the final phenotype of a given trait. And best example is the skin color. So skin color in humans, many genes contribute to the skin color. So how does polygenic inheritance work? Each gene in the group of polygenic genes which control a trait like skin color, each of these genes plays a small role in expression of the trait. So you can see these are different variations of the brown color or the skin tones we have. We have fair, we have medium brown, and we have dark. So different genes code for these colors and they express a part of this trait, the whole skin color trait. This allows for a trait to vary greatly from person to person. So we see from populations across the world, the skin color varies greatly. Height is a polygenic trait, which is why people can be very short, very tall, or anything in between. So height is also a polygenic trait. And frequency of the phenotype, if you plot it, it comes somewhat like this. So the left extremes and the right extreme, if you measure, let's say we consider height as a polygenic trait, the left ones will be less height, lesser than 4 feet, and the right ones will be more than 6 feet. But generally, the average height people will be more in numbers, higher frequency, around 5 to 6 feet, will be the higher frequency of individuals in the center. So this will give a characteristic bell curve. So going on with the definition, a polygenic trait is a trait that is coded for by many different genes, poly and genic. With polygenic traits, there is no set number of genes that could code for a specific trait. So a specific trait is brought upon by many genes. Instead of AA, BB, and CC, this type of trait would show all the genes working together like AA, BB, CC. So all these genes work together towards a single phenotype or a single trait. That's why it's called polygenic inheritance. Now, the distribution of polygenic traits that follow this pattern, the traits which follow this polygenic inheritance patterns tend to make a bell-shaped curved graph, where we saw most of the common or average cases will have the highest frequency, whereas there are rare cases in at the both ends of the bell curve, extreme phenotype at the both the ends, and the average phenotype with highest frequency. What this means is that there will be large number of cases where people have a medium value. So people will have more average value. Let's look at height again as an example. So again we take height as an example. The further we move away from the middle, more extreme height becomes. So on the x-axis this is increasing height. So the further we move away from middle, the more extreme the height becomes. And as we move to the left, people get shorter and shorter. And as we move to the right, people become super tall. So these yellow regions are rare cases where people are super tall, height greater than 6 feet. And these on the left hand side of the bell curve are rare cases where people are lesser than 4 feet. And most of the average height is 4 feet within 6 feet. And if you plot this median, then this would be around 5 feet 5 inches. So the polygenic traits have this bell-shaped curve characteristic where the average value or the population adopts a medium value with little extremes at the ends. So the answer was answered correctly by Simontini. Bell-shaped curve is shown by polygenic inheritance. Now, is this clear to the students? Polygenic inheritance and how a population will show polygenic inheritance, the basic fundamental definition, and how does these traits work? Is it clear to the students? Just let me know in the chat if it's clear to you guys, then we can move on to the next question. Okay, good, great. Now let's see the next question. You might require pen and paper for the next two questions. We have already reached the last, towards the end of these sessions. We have two more questions to go, and thereafter we'll wrap up this session. So I'll encourage all of you guys to at least try to solve these last questions. These are very important, and also it will be great fun if you get it right? So I urge you all to answer these questions. Question number 9. So this is a genetic cross now, cross-based question. A female red-eyed fly was crossed with a white-eyed male. So read the question carefully. So what is the starting generation? I hope you can see my slides changing. A female red-eyed fly was crossed with a white-eyed male. So the female is red-eyed, male is white-eyed. Now what happens to them? Half of the females produced were red, the other half were white. So out of the female progenies, half of them were red, half of them were white. Half of the male produced were red, and the other half of the male produced were white. So if you jot down, you see that half of the males are white, half of the females are white, half of the males are red, and half of the females are red. Then what is the genotype of the female parent? So we are here to find out what genotype the female parent must carry, so that when mated, when crossed with a white-eyed male, will produce offsprings with certain frequencies, such as half of the females were red, half of the males are red, half of the females are white, and half of the males are white. I know it might take some time for you guys to jot it down and calculate and find out the genotype of the female parent. Let's take a little more time and ponder about this question. And the options are W plus W plus W plus W. For your information, W plus means red and W is white. So if you remember, the red eye is a dominant phenotype and white eye is a recessive phenotype in Drosophila. So W plus is the dominant allele and W is the white-eyed recessive allele. And Y is the Y chromosome of male. So in Drosophila we have XX as female and XY as male. So similarly, the options are W plus W plus, option A, option B, W plus W, option C, W plus Y, option D, WY, option E, WW. You have to remember, you have to find out what is the genotype of the female parent. Female parent which was red-eyed and the male parent which was white-eyed. Now what genotype the female parent should have to produce progeny frequencies in which half of each gender harbors the trait of both father and the mother. You try to solve it. I'll wait for some more time because this question is a little tricky. I'll wait for some more time and leave you with the question and the slides. You try to solve it and come up with an answer. Answer in the comment box, mention in the comment box what you think is the correct option. Okay, Rahul has also already solved and he's answering option A. Very good. W plus, W plus. Okay, you tried. And let us wait for some more answers. Simonthini is answering C. Pushpanja is also answering C. Good, good, you guys are trying. So Rahul has option A. Two people, Simonthini and Pushpanja, are answering option C. Now let us look at what is the correct answer, the moment of truth. The correct answer is option B. W plus W. Pushpanja and Simonthini, you were at the right answer, but you failed to read the question properly. So the question is, what is the genotype of a female parent? So option C is a male parent, right? Because it has a Y chromosome. Option C cannot be the genotype of a female parent. So you could have eliminated the answer, but the answer correct option is option B. I'm sorry to Rahul also, his answer was not correct. But let us see how we reached to this answer. So all these options with Y chromosome will not be the answer because the question is about what is the genotype of the female parent. We do not need the genotype of the male parent, right? And C is not the correct option because W plus is a dominant allele for red color. With the Y chromosome, the male parent will be red-eyed. So it can never be the answer. We have a white-eyed male as a parent. White-eyed male will always be W and Y. But that is not the question. The question is about what is the genotype of the female parent. Now let us go slowly, progressively. So what are the possible genotypes of these parents? So as I said, the male with the white eye can only have one genotype. White-eyed male will have only one genotype, which is W and the Y chromosome. Whereas the female can have two kinds of genotype. Either two W plus, two dominant alleles of red, or heterozygous, where one dominant allele of red and one recessive allele of white. And males are therefore called hemizygous because they only have one allele for X-linked characteristic. Hemizygosity makes the description of dominance and recessiveness irrelevant of XY males because each male only has one copy of the gene. Since males have only one copy of X, there is no concept of dominant and recessive because dominant over what? Dominant over the other gene of the X chromosome, right? But since males have only one, males are said to be hemizygous. Now what happens if we consider this possibility where the female is homozygous for red dominant allele? Now if the female is homozygous, if we separate it into gametes, the eggs of the female will have both the red allele, W plus allele, and the sperm will have recessive white allele and the Y chromosome, right? Because the male parent is white-eyed, and if this is the case, if the female is homozygous, then we have W plus and W plus. Now if we draw the Punnett square, we see the daughters that will form from the X chromosome and the X chromosome. Daughters are red-eyed and heterozygous, so all daughters are red-eyed, while all the sons are red-eyed and are hemizygous. So the Y chromosome from the sperm will fuse with the X chromosome of the eggs carrying the dominant allele, and thereby these males will be hemizygous dominant. So males will have red eyes and the female will also have red eyes. So could you see? So this is not an option. So this cannot be the female parent's genotype because if we have this kind of if we have this kind of genotypic distribution, we are generating all the daughters to be red and all the sons to be red-eyed. Now what happens when our female is heterozygous? When our female is heterozygous, when the female is heterozygous, W plus is the dominant allele for red color, W is the recessive allele for white color. When the female is heterozygous, now we have eggs with a red allele, W plus dominant allele, and a white allele, which is recessive. And we have sperm from the white-eyed male parent, which has recessive white allele and a Y chromosome, right? Now look at this Punnett square carefully. W and W plus will give a red female since this is the X chromosome. W and W will give a white female since this is the X chromosome and this is the X of the sperm. Now in the males, we'll have one proportion of red male and a white male. So this is the condition that was given in the question. We have red females as well as red males with white females as well as white males. So the answer to this question will be heterozygous female, which is W plus W. So this will be the answer. Option B is the answer, W plus W. So a heterozygous red-eyed female when crossed with a white-eyed male will produce one half females red, one half females white, one half males red as well as one half males white, as we can see in this Punnett square. Is it clear to the students? This was a tricky question and it requires you drawing on to the Punnett square and exploring all the possibilities. I hope the crosses and everything was clear to the students so they could understand. If yes, just let me know. Why is the female heterozygous in this question? because only when a female is heterozygous and the male parent is white-eyed with a recessive allele, we can have four different factions based on gender. So female's spectrum will be red and white-eyed, while male spectrum will also be red and white-eyed. Is it clear to the students? Yes. Good. Cool. Now let's move on to the last question. Now this is also a question where you would need pen and paper. So I would give you a much longer time to think about the answer. And this is the last question, my friends. So I would urge everyone, I would expect all five of you to answer. So I think we are five people here, five or six people. I would expect all of you to answer. This is a difficult question, but once you sit down and understand the question properly, you will be able to solve out the answer. And don't worry if your answer is wrong. I'll let you know what is the correct answer and solve it for you, and we'll solve it together so that we can understand. And when we face such a problem in our assignments and in our exams, we will be able to solve it, right? So there are two genes, A and B, which were found to be tightly linked in an organism. So you have to read every part of the question very carefully. A parent homozygous for the dominant alleles of both the genes was crossed with a parent homozygous for recessive alleles of both the genes. So there you have two parents, a parent homozygous for the dominant alleles of both the genes and a parent recessive homozygous for the both the genes, right? And then we have F1 generation. Now the progeny of F1 generation were allowed to self-fertilize and F2 progeny were obtained. So then they went for F1 generation cross. So F1's male and female are self-fertilization and progeny was obtained. F2 progeny was obtained. Now the question is, what would be the phenotypic ratio among the F2 progeny? So once you self-fertilize two F1 and you get F2 progeny, what is the phenotypic ratio among the F2 progeny? Option A, 3 is to 1. Option B, 1 is to 2 is to 1. Option C, 9 is to 3 is to 3 is to 1. Option D, no exact ratio can be determined as genes are linked. Students, take your time easily, go through the question yourself and try to solve it for the answer. What do you think is the phenotypic ratio in the F2 generation? And I would expect all five of you to answer whatever you think is the correct answer. Pushpanjay has answered option B. Okay, nice try. And others, Ashwin, Simontini, Rahul. Simontini is answering option D. And Rahul has answered option A. Ashwin, can I expect an answer from you? Ashwini, are you able to follow these sessions, these questions and explanations? Were you able to go through this last 10th question? Then you could answer it easily, whatever you think is the correct answer. Okay, let's not delay it any further, and let me reveal the correct answer. The correct answer to this question is option A, 3:1. So Simontini answered D, no exact ratio can be determined as genes are linked. I know they are tightly linked genes, but even tightly linked genes can have a recombination frequency. And we would see that recombination is not the event that is governing the phenotypic ratio. Now let me solve. So I have solved this question on pen and paper for you guys myself and uploaded my notebook images. So if you follow it with my cursor, what is given in the question? Given in the question are two genes A and B which are tightly linked. And what do they mean when they say tightly linked? They always assort together. So always A and B will assort together. A and B will inherit together into a gamete. So that is the key to the problem. Now the first given question is the parents. So one of the parents is homozygous for dominant allele. So the genotype will be AABB. Do you agree with me? Do you agree with me? AABB. And the other parent is homozygous for recessive allele, small AABB. Do you agree with me, guys? This is what is given in the question. Yes, good. Now we draw what are the gametes that will come from these parents. So if you represent AABB like this on chromosomes, so AB are two tightly linked genes, and we have AABB and small b in this arrangement. Now if you make gametes, these undergo meiosis, the gametes will be capital AB from one parent, and small ab from the second parent. And these are the gametes for F1 generation, the first cross. Now if you cross them, these gametes, they will produce only one type of progeny, which will have AB and small b chromosomes, right? And this will be a heterozygous progeny. So F1 will be heterozygous with one AB from this parent and one small b from this parent. Do you agree with me, guys? Because I want to go step by step in this problem so that you guys will feel assured that you can solve these kind of problems. Yeah, great. And now we cross two F1 progenies. So we know one F1 progeny is heterozygous, and we have similar kind of another F1 progeny, AB and small b, right? A and A are alleles of the same locus, B and small b are the alleles of same locus. Now in this parent, if we take out the gametes, we'll have A and B together, because A and B are tightly linked. They will always inherit together. And small ab will also inherit together because they are also tightly linked. Because these two locuses are tightly linked. So we will have a gamete with capital A, capital B, and another gamete with small b. And similarly for the other parent. Now if we do combination when we have progeny, there can be four probabilistic progenies where this gamete will fuse with capital AB to form dominant phenotype AABB. This is the parent phenotype when we started first. This AB can also fuse with this small ab to produce a heterozygous but dominant phenotype, and then this small ab will fuse with AB, capital AB, to produce another heterozygous dominant phenotype, and then small ab can produce a recessive phenotype. So based on phenotypic ratio, all these three progenies will be dominant because capital A is dominant over small a and capital B is dominant over small b, while only one of these will be recessive. So the answer phenotypic ratio will be 3:1. So this is the correct answer. I see somebody has answered option B, 1:2:1. I think Pushpanjay has answered option B. Pushpanjay, you were slightly correct, but 1 is to 2 is to 1 is the genotypic ratio. So if you see, what do we mean by genotype and phenotype? Phenotype is the expressible characteristics. So the phenotype will be dominant. The capital A will dominate over capital small a and capital B over small b. But genotypically, there are three different genotypes. One will be capital AB AB, the other will be heterozygous, capital A, capital B, small b, which will be 2 in number, and the last one will be small a, small b, small b. So if the question would have asked about the genotypic ratio, then the answer would have been 1 is to 2 is to 1. But the question has asked about the phenotypic ratio, how the genes will express. So then the answer would be 3 is to 1, which I think Rahul has answered very correctly. So dominant phenotype is to recessive phenotype. Now, is this clear to the students? I have worked this problem out for you guys on pen and paper myself so that it's easy for you guys to understand. Do not mind my bad handwriting, but it's important that you guys understand. Is it clear to the students? Are you able to understand this, how we solved this question? Two tightly linked genes, and what is the dominant, what is the phenotypic ratio? Yes, great, great. Fine, and with this we come to the end of this session. We have solved 10 questions corresponding to the lecture week 3, and I'll just stop my presentation, stop sharing my screen. Yeah, and I hope people were able to follow at least one or the other concepts here, and you were able to revise the kind of problems that you are going to face in your assignments and in the exams. I hope it is clearer, and I encourage you all to attend all these problem solving sessions where you can have direct interaction with the TAs who will help you to understand this course better, and also urge your friends who are enrolled in this course, if you have, to join the TA sessions. I will assure you that these will be very fruitful and helpful, and I hope you have learned something. As mentioned earlier, now what I'll do is I'll just open my YouTube link and copy paste it for you guys, so that will have the recordings of the earlier session as well as today's session and all the upcoming sessions that we are going to have together as a problem practice, right? And via NPTEL you must have already received the link for Google Drive containing access to the slides of this presentation. So let me just give me a minute, and I'll just upload the playlist link. So you will find all the videos here. I'm just pasting it here in the chat box. So this is the link. You can find these videos, or you can just type my name, Ashutosh Behera, and NPTEL problem solving sessions, Introduction to Developmental Biology. You can get it. Also in the course tab, I think when you entered Swayam course tab, you can find it there, and you must be getting emails from NPTEL team regarding the drive link for slides as well as recorded sessions. So I hope you're able to learn something from this recorded session, and it is a two-way street. Even I am learning many things while teaching you guys, and hope to see you soon next week, where we'll be solving practice problem corresponding to lecture week series 4. So be prepared, and take care. Bye bye. I'll end this session. You might be interested in copying the YouTube link, and thank you for joining.